We're going to go, we've been talking about the present. We're going to do a poll that talks a little bit about looking into the future. So we're going to post a poll about predictive modeling. Is predictive modeling, like Nowcast, used for water quality notification in your recreational water? So we'll take about 30 seconds to work on that. And while those data are being collected, I will point out that our next panelist is, or our next session director, is Tracy Bone from EPA. And she is not only a session leader, but she's really been the engine behind getting this conference together. So here are the results. And I'm going to hand this over to Tracy so that she can introduce her panel and talk about how the results intersect with today's conversation.
Thank you, Steve. Hello, everyone, and welcome to the second half of session 2, Advances in Monitoring. It looks like there's not a whole lot of predictive modeling going on across the country. So maybe what you hear from my panelists will encourage you to look into it, because it can be a great tool. So I'm going to introduce each one of my 4 great scientists one at a time and ask them a question. And so if you guys can promote them as I introduce them. The first scientist I'd like to introduce is Dr. Anis Blanch from University of Barcelona. He's a full professor of microbiology. His research focuses on bacterial methods, MST, and bacteriophage. His presentation, that hopefully you have already viewed, is titled Predictive Models and the Fast Detection of Colophages for a Paradigmatic Improvement in Rapid Assessment of Water Quality. Hello, Dr. Blanch.
You're still muted.
As he unmutes, I'm going to tell you that you described in your paper about bacterial monitoring and forecasting software, or as we like to call it, predictive models, that you used for risk assessment at your beaches in Barcelona. You described the work you did to find a faster monitoring protocol for lifting the beach when a beach is closed. So as we all know, bacterial tests take about 24 hours to get the results back. But also by your research and by many others, often the contamination at the beach is gone within 2 days, if less. Not all beaches, not all contamination, but often. So your research shows that you can rely on a 4-hour reading of somatic colophage to predict the beach is safe to reopen. So based on your comparison of the bacterial culture results with the colophage results, I wonder, did you also compare any modeling predictions with the colophage results in the bacteria? And if those two diverged, what did you do?
Okay, thanks. Thanks to me and to give me the opportunity to be here with you today. And I'll be glad to answer your question. Yes, we did. In our case, and with 229 cases, it was compared with the results of coliphages and the bacterial indicators. I pointed out that complementing bathing waters analysis with somatic coliphages not only gives us complementary information as viral indicators for risk assessment, but can also allow us to advance results either by models like the one presented, or by some of the new tools for rapid analysis of coliphages. However, at the moment, it will be interesting that the incorporation of coliphages as viral indicators in bathing water will us allow to have more information to be able to make more robust and fast decisions regarding the reopening of recreational waters. Particularly, we in the analyzed samples only found 2% of disagreement with bacterial indicators. So we find bacterial indicators positive, but coliphages were below our cut-off, that was 180 PFUs. If we have a bigger number of cases, then we could adjust the cut-off value that we have now. And our beach, as I say, it's 180 PFUs. So that means that, yes, we check it with coliphages with bacterial indicators, but this cut-off value should be determined for each particular recreational area. to have validated your model. I don't know,
Very
did that give you an answer?
Yes, it's a great answer. And we're very interested in colophage EPA as well. And there's additional talks in session 6. So for the moment, though, let me introduce my second panelist, Dr. Sarah Bickman. Dr. Bickman is a senior scientist and the product manager of the Water and Food Division at Light Deck Diagnostics. Her presentation is titled Portable System for Early Detection of Harmful Algal Bloom Toxins. Dr. Bickman should appear any second now.
Connection.
Hi, great. So, Dr. Bickman, your new technology is very interesting. And so, as you know, the concern for and recognition of HABs seems to be growing across the United States. I wanted to ask you about the sample prep step. So it seems to me that your technology was first designed perhaps for drinking water, but you've also included preps in protocol for testing and then also dilution step for rec waters. So I wonder if you could tell me a little bit more about this technology in recreational waters.
Yes, thanks. Thanks for the great question, and thanks to everybody for doing all of the organization for this conference. So yes, you're right, that sample preparation is something that we've thought quite a bit about, and we deliberately did not supply a lot of information in the presentation. And the reason for that is that we do think that there are options. Just as you implied, there are options for measuring it around the drinking water limits for these toxins. And then there are also options to take your sample and then do a dilution step and then measure much closer to the recreational water guidelines. So that's the reason that we didn't include that. The other issue with sample preparation that we've put quite a bit of thought and work into is lysis. So basically what happens with many of these species that create the toxins is that they contain the toxins within the cell walls. You would want to lyse or break apart the cell walls to release those toxins. The reason that we haven't included that information in the presentation is that there are really 3 options. One option is that some people prefer to measure just the toxins that are dissolved in the water, in which case you can take your sample and go directly onto the test. A second option is that you can lyse the samples by doing the 3 freezing and thawing cycles that's recommended by the US EPA method 546. And so that's a really good option if you do want to lyse cells. And then finally, we've been working on our own portable lysis method that would be more rapid and could be used in the field. That's something that we've put quite a bit of effort into. It's still under development. We think it's very promising, but it's not quite at the stage where we're ready to roll it out to have everybody using it.
Oh, that's very interesting. And it certainly falls in our session of advances in monitoring. I think we're all struggling a bit trying to figure out what to do with HABs and cyanotoxins, and there are also more discussions and more papers in other sessions on HABs. So it'll be interesting to see what comes of your new technology. But let me bring in our third speaker. Deneen Blackwood is a research specialist with the University of North Carolina at Chapel Hill. She has worked in the fields of molecular and environmental microbiology for over 20 years. And her presentation is titled Use of Novel Autonomous Technology for Improved Water Quality Monitoring in High Priority Recreational and Shellfish Harvesting Waters. Shannon Briggs has already mentioned her cute little boats, and I desperately want one, but hopefully we can see Deneen here shortly. Because I want to ask her about the work she did in Beaufort, North Carolina. The first thing I need to check is whether it's pronounced Beaufort or Beaufort, North Carolina.
It's Beaufort. Yeah.
Thank you. Thank you. So your research, it sounds like a lot of water shared in a lot of water quality concerns is shared among many coastal cities and towns around the US. So many marinas worry about fecal contamination and stormwater runoff and coastal development and aging infrastructure. And so your autonomous technology boats, they expand monitoring options to allow in-system or water monitoring, which provides more information than just land-based monitoring ever could. So I wonder if the results from your measurements for dissolved oxygen and Vibrio and nitric oxide and NSG, you monitored a great deal. It's very fascinating. Have those results been looked at by local water managers and perhaps led to actions or planning to somehow reduce DO and so forth? And also, what is the reaction from local boaters when they see your little autonomous boats whizzing around the marina?
Great question. So this work was supported in part by a NOAA NEARS grant that was looking at stormwater impacts on Rachel Carson Estuarine Reserve, which is an adjacent water body to some of the sites that you saw in the presentation. And the work has supported multiple master's, PhD students, and numerous publications. Also, one of the collaborators on our project is a PhD candidate at Duke Marine Lab. He's also the mayor of the town of Beaufort. And so he was able to leverage a lot of support by the town for our work. And we also, Stormwater Advisory Committee was established. And there's lots of, in Beaufort, there's lots of stakeholders, other universities like NC State, Duke, there's the Coastal Federation, there's all these, stakeholders that are interested in good water quality, of course. And so they form part of the Stormwater Advisory Committee. So some of the outcomes from this work is actually leveraged about $30 million to the town for infrastructure repair. As far as safety concerns, because we have all of these different marine labs in the area, there's a lot of things like that that go on anyway. And we are also home to several marine bases, which have all kinds of covert operations going on. So with all the graduate and scientists there, it's not like, a really big deal to see kind of things like that all the time. So.
Very interesting. Well, I hope to get down there and maybe I'll see it whizzing by one day, because I think that we all have to expand to some more offshore monitoring to really figure out what's going on at our beaches.
Yeah.
Okay. We've kept Dr. Joshua Steele hidden for long enough, so I'd like to introduce him. He's our last speaker, our last panelist. He's a microbiologist at the Southern California Coastal Water Research Project. I always want to say program, SQUIRP. And he specializes in environmental microbiology and microbial ecology, including cultivation independent tracking of pathogens and natural microorganisms. And the title of Dr. Steele's presentation is Antimicrobial Resistance in Wastewater Effluent Streams, Discharging to Urban Coastal Waters. Hi, Joshua. So you're looking at antimicrobial resistant microorganisms, a very big issue these days in wastewater and in outfalls. And you found that there's a similarity in the influent signature to wastewater treatment plants, so that you could even track, where it was from and identify it. Were you able to look at the influent sewage signature for the Mexican wastewater treatment plant that you were looking at and comparing in your study? And would you expect that similarity? And and where do you think we're going to go? How would you see this ability to track and identify or fingerprint sewage upcurrents? From how would we use that information?
Thanks, Tracy. That's a great question. Or set of questions.
Yes.
We did look at the influent across multiple plants, and one of the plants we were able to capture was the International Wastewater Treatment Plant, which does have influent that is split. Some of it goes to that plant, and some of it goes to another plant a little bit down coast in Mexico. And so we did have influent that was shared between those plants that we were able to measure. And so yeah, we did. we were able to measure that influent. And the interesting thing is the influent signatures were remarkably similar, whether it was the influent in Mexico or throughout the different Southern California plants we looked at. But the effluents were distinct. So that was where it was a really useful tool. Once it's gone through the treatment process, then you have an effluent that you could track to that plant. Probably you could say, well, I know where this came from based on how this looks. And that's how we were able to look at the effluent from that from the Trent that was down current when the south swell came up and brought it up. So the way we envision this going is this is yet another tool to use for source tracking. And you can we have sort of the really broad-based tools such as the fecal indicator bacteria that give you a really that catch everything under the sun that they can and give you a result of whether it's contaminated. We have the more specific tools like the microbial source tracking markers, where you can distinguish human and dog and gull and those, which do a great job. And then this would give you sort of one more level of ability to distinguish things, the sequencing or some of these other, where you can say, well, what type of human is it? Is it coming from a sewage treatment plant, or is it coming from a leaking sewer pipe, or somewhere else? And so that's sort of how we're looking at using this. And the one layer on top of that we had is sort of bringing back in the quantitative results, looking at the antibiotic resistance genes. Then you can say, okay, well, now I know where it's coming from, but are there other threats, other risks as a part of this besides what we are typically looking for?
Yes, I find it fascinating, and I think that is the logical next step, which will be talked about in the remediation session. To fix the problem, you've got to figure out whether it's a septic tank or a wastewater treatment plant, or it's a cattle farm. It's all very interesting. But then once you get even that subset of information, where do I go with it then? So sounds like a very interesting tool. So I thought I'd then, you all are on the cusp of research and information and technology. So I thought I'd just sort of throw open the million-dollar question. Where do you see monitoring recreational waters go in the future? Or what research would you like to see, or do you plan to do yourselves next? So Joshua, we kept you waiting. Would you like to start first?
Sure. So I mean, I think the research I'd like to see next, or where it could go next, is sort of looking, taking these sequencing tools one step further and looking, again, broadening it back out. So use it as a screening tool for different genes, different risk factors that we don't currently monitor. And I think that would be something that would be very useful, both in terms of antibiotic resistance, but also in terms of pathogens, sort of a broad screening type tool. With antibiotic resistance in particular, looking beyond just the ones that we are concerned about for human risk, but also concern for marine mammals, or even endemic bacterial communities that may be taking these up, like the Vibrio communities in some areas that may be taking these genes up through gene transfer and then transferring that risk to people through sort of circuitous route. And that would be, those are both places I think that we need to go to next.
Absolutely. Yeah, Vibrio's moving up the coast, as they say. So let's go to Deneen and ask you that question. I actually also had a Q&A pop up asking, have you figured out how to equip the autonomous sampler to collect a water sample for lab analysis? But feel free to answer whatever question you'd like. Except you're muted, so you have to hit the button. I know.
Sorry. So no, we have not. So one of the previous versions of the autonomous boat was actually manned just to be able to collect the sample. However, the coordinates could be programmed into, the technology. So we have not figured out how. We have not been able to equip the boat with a sipper or an auto sampler, but I do not think that would be a problem. We might have to have a bigger iteration of the boat in the future. So as far as, where I would like to see the monitoring going, I think one of the hurdles is that, the EPA methods, they're quite cumbersome in their lengthy, sorry, Oren, but, and it would be nice if we can have like, a more standardized, streamlined technology or methods, because we all kind of are really familiar with digital PCR and qPCR. So if we could focus on maybe streamlining the methods so that more people would accept them without having to, go through so many pages and pages and pages of, the methods, which can be overwhelming. And then also try to find more support to train the next workforce in water quality management using these, streamlined methods. And we could find maybe a good blend of culture and qPCR methods, because, as Shannon talked about, they're not exactly perfect, but they do work pretty well in most locations.
Well, that absolutely a combination of the methods, and I know Oren is working on everything, and we'll make sure he gets to work even harder. No, just poking Oren a little bit there.
Yeah.
So Sarah, what would you like to, what are you looking ahead for in monitoring?
Yeah, I think there are really two areas that algal toxin monitoring needs to expand in. One is that a previous panelist noted this, but every time the wind blows or the time of day changes or the sunlight changes, the amount of toxins can change significantly. So I think we need to do more monitoring in more places at more times. So I think that's a huge area. Another area that I think would be really interesting to expand upon is monitoring for more toxins. So right now, it seems like most people are monitoring for 1 or 2 toxins that are common to their area. But I've spoken to a number of people lately who say that suddenly they've started looking for a new toxin, and lo and behold, they have it in their local areas. So I think we need to expand monitoring for more toxins in more locations at more times. This is something that we've been really lucky to be funded through NOAA for, is that we're working on expanding to monitoring 4 toxins instead of 2. But I think all technology platforms are moving in that direction of having more capabilities.
And so monitoring for multiple toxins in the same sample, which also can give you a picture of the ecosystem. I don't know if that's the right word, but it gives you a more complex picture of what's going on in the waters.
Yes, thanks.
Absolutely. I absolutely. Anisette, so what's on your wish list in Spain?
Yeah, I agree with some of the comments here in this panel and in the previous panel about the use of bacterial indicators, mainly E. coli and Enterococci, for the monitoring of recreational waters in recent decades. They have provided very good results to support the health, safety, and reduce the risk of outbreaks. But I think we still need to make a small analytical effort to increase the safety of our recreational waters. And probably that will help us to decrease the outbreaks that still occur, even when the bacterial indicators are correct, are fitting in the regulations. Yesterday, we have some presentations from Sam Dorevich that was telling us that still there are many cases, not just in the States, also in Europe, it happened. So in that sense, I think that was the approach of our work about the incorporation of colifages as viral indicators to try to control or monitor those outbreaks produced by waterborne viruses. I think this is one of the ideas that we have in our hands, the technology. So it's just to try to monitor and to follow what is the behavior of these viruses. Another thing, additionally, has been also commented by my colleagues, the new technological approaches and fast methods, not just for bacterial indicators, also for viral indicators, could contribute significantly on the new vision of a smart water management. We need to provide from the microbiology digitalized results that will help us to develop modeling, predictions, and so on, and integrate all the data. And we cannot forget our colleagues in engineering that we need also to make close to the analysis of the results. And I think digitalization should be a challenge for all the microbiologists.
Absolutely. Predictive modeling, even though it didn't rank too highly on our poll at the beginning of the session, I think it's picking up steam, and all those inputs that you mentioned and the extra expertise from engineers to help answer some of the questions would be great. I have a question for you, Dr. Blanc. It is, at the locations where you found good agreement between coliphage and traditional fecal indicators, could you describe the type of wastewater treatment for the effluent affecting those beaches? In other words, where you found a good agreement, was there chlorine disinfection of the effluent? That would probably affect the coliphage versus bacterial results.
Yeah, well, that's a good question, because we develop our study in the Barcelona beaches. So that means we have 9-10 beaches, that depends of the year, in the shoreline of Barcelona city. And there are two big wastewater treatment plants in Barcelona. They are working for 1.5 million of inhabitants. And they are working in secondary treatments, but as you probably know, in Europe, it's not regulated to treat the effluent of the secondary treatments before we are disposal in the environment. So it's a secondary treatment. The effluent of the secondary treatment goes through the under the sea and goes several kilometers into the sea. And it's not influencing our area. So the quality of the beaches is excellent. It's 6 million of bathers every year in the season. So the quality has to be very well controlled and monitoring. And this is why the municipality developed prediction of these beaches. But the direct answer is yes, we have two big treatment plants with secondary treatments. They are working with tertiary treatments to recover this water for recovery our water resources, but right now goes directly to the sea, but several kilometers into the sea.
Thank you very much. That's very interesting. That's good to know. Well, so my panel represented the two ends. I had California and Spain, so I think we geographically nailed it. And I thank you all. I want to thank you very much. I know taking this conference workshop online was a lot of extra work for everybody, and everybody made their presentations and sent them in, and I appreciate that, and I appreciate for showing up today. That was great. I learned a lot.
Okay.
Steve, should I give it back to you?
Sure.
Or
Thanks,
do I have to talk
Tracy.
for one more minute?
Nope, you can hand it back, and we will use that time appropriately.